So welcome everyone to MSQ webinar. Today I'm so honored to have Dr. Jacob Glanville to join me, and he is the president and the co-founder of Distributed Bio, and he is also computer immune engineer, and he also featured in Netflix doc series Pandemic. That's where I knew you. And welcome, Dr. Glanville. How are you today? How's your day look like today?
Thank you for having me on. I appreciate your time. It's been busy. My team has been working in shifts, morning and evening, to work forward to finish the engineering of this molecule and to plan the next steps once the molecule is ready. So it's been a mixture of work here in the laboratory, meetings with other partners, companies, the military, and just pushing this thing forward.
That's great. Thank you so much. I think you are in your lab now, right? Actually, now is 6 p.m. in California.
Yeah. So my team has been coming in shifts. So even since the shutdown, they've been coming in. They started about 6:30, 7 a.m. and then they often in by like 2 a.m. in the morning. So we actually are working so much, we're running into problems where in the middle of the night, the cells sometimes don't have enough time to divide by the morning. So we're coming up with adaptations. So yeah, we're cranking away.
Wow. So after watching Pandemic, I was very intrigued by what you are working on. Can you share with the audience what Distributed Bio is trying to achieve?
Sure, thank you. So Distributed Bio is a computational immunoengineering group. We are surfing on some of the advances in high throughput data acquisition and high throughput engineering robotics to build better populations of antibodies. So we are specialists in creating antibody therapeutics, discovery and engineering, and we're also specialists in eliciting antibodies in animals, so vaccines. The company really is two parts. We never took on venture capital. So since 2012, when we launched, we had a software platform that allowed the analysis of hundreds of millions of antibodies and T-cell receptors. And that platform was used by many companies, and it was run on the Amazon cloud. So that enabled us to have many partners like Pfizer and Boehringer and Ingelheim and Gilead and many others. But it also gave us the data to help understand how does a human body produce excellent antibodies. And so given those insights, we began building physical technologies. So antibody discovery technologies to be able to go and make antibodies as drugs against challenging targets like GPCRs, ion channels, peptide MHC complexes, and other targets like viral targets. So the company, as it grew, we perform contract research. So companies come to us and say, hey, we want to make a drug against a plaque in the brain or a receptor on the heart. And we do that work with them, and we're paid for it. And we have milestones and a small royalty. That relationship has worked great for us. We've grown quite a bit. And then the unique thing about Distributed Bio is because we have some of these unique tools and technologies, I take ideas that I think are really cool, that I think are sort of a low-hanging fruit because of the new golden age of biotechnology, but are so risky, I would not want to offer it as a service to others, but I'm willing to test it internally. And I use some of the profits of the company to go run those research projects. Not all of them work. Some of them we try and we decide to abandon them, but some of them have been remarkably successful. So one of them is featured on the Pandemic Netflix documentary, and it also won a Gates Foundation Grand Challenge in the Pandemic Threat Award. And that's a technology for creating a broad spectrum vaccine. We're going after influenza initially to make like a universal vaccine, a single shot that could protect against at least the next 20 years of influenza. And that technology is working quite well. And we are moving into first the veterinary market, so pigs, where it's about $175, $180 million a year for pig flu shots. And you can access that market in a couple years. And we're about to run the in vivo live challenge studies, which would be enough to initiate that. And then a few years after that, after phase trials, you have the human vaccine. And in that same technology, we are in the early stages of testing it against lentivirus, so HIV, and a couple other pathogens. But at this point, the data proof is all around influenza. We also have a couple assets that are monoclonals or polyclonals. So we work on a fully human broad spectrum antivenom for snakes. And that technology really emerged from, we found a guy who spent 17 years self-administering snake venom from taipan, cobra, mamba, a series of snakes, and he'd built up hyperimmunity. So we took his blood, analyzed his sequences, and have found a series of antibodies from his blood that recognize and neutralize like neurotoxins and all snake venom and so forth. So that's another technology, and that's supported by the National Institute of Health here in the United States. And then we have also the relationships with USAMRIID, NMRC, and RARE. So these are departments of the US government and military that provide support for anti-pathogen research. And as part of that, I think the most interesting one right now is that since January 25th, we have been developing an anti-COVID-19 monoclonal or maybe even oligoclonal therapy. We're still deciding how many antibodies go in. But this is a treatment of, we're still deciding, 1 to 3 antibodies that are neutralizing, where you could infuse into a patient, and unlike a vaccine, it will work immediately, and it will work on elderly and the immunocompromised, where the vaccine maybe won't work that well for them. It will work within 20 minutes, as opposed to a vaccine that may take 6 or 7 weeks to become effective. There are some advantages to that. So we're making that as a medicine. We are doing everything in our power to move this as quickly forward as we can. And I have some slides describing the progress of that project.
That's really wonderful. So I think all the, what you're doing are very fascinating. And so for the universal vaccines, what's the progress now? What's the status now?
Yeah, so the status is we've run 3 animal studies, and we've been able to demonstrate that pigs who receive the vaccine can bind and neutralize viruses back to 1918 and into the future. Where we are right now is the Gates Foundation gave us an award. It's an initial set of $2 million with then $10 million of follow-on. Thank you regarding the hair. I saw a comment. And the current studies are we are going to run live challenge studies. So this is where we give pigs and ferrets our vaccine, or we give them a standard bivalent vaccine. And then afterwards, we're going to challenge them with a heterologous future virus. So we're going to pretend it's 2009 when we create our formulation. We'll only use variation and information from earlier than 2009. And then we will challenge them with a future 2015 strain that happened after the pandemic shift of H1N1. And that lets us know, is it able to neutralize? Is it working better than a standard vaccine toward it? And is it providing vigorous protection? For the pigs, that's really all we need. At that point, we can start going into the markets. So working with clinical markets. So we're talking to Merck Animal Health and Zoetis, and we're balancing the desire to produce the medicine ourselves as opposed to just do a licensing relationship with an existing veterinary company. I think we're flexible there. Mm-hmm. I understand there are a lot of pigs in China. So as long as when that live challenge looks good, we'd certainly be interested in talking with organizations in China as well. And then on the ferret side, to get a vaccine approved for humans, you don't really need to do non-human primates. You do ferrets. Ferrets are required for historical reasons. So once the ferret study shows live challenge protection, at that point we would launch a phase 1 study in humans.
Mm-hmm. Actually, I find it's very cute from there's cute part of the scientist and about you, and also the movie Pandemic is you build do the trial for the pigs back to your hometown. And that is very I just feel it's very amazing. And also you are really think outside of the box to save the cost, and that's why you never take the financial investment for your company.
Yeah, I think it was, I'm always willing to ask, just because other people do it a certain way, is that really what needs to be done? And with vaccine science, you do need to do animal research. There's not a lot you can learn. The immune system is so complex as the adaptive and the innate and how it interacts because even each organism is relatively different. I think there's only so much you can learn in a test tube, in a petri dish. You have to go into an organism. And I didn't want to go into mice because there's no market for flu shots for mice. I can make the best universal vaccine for a mouse and nobody cares. Whereas pigs, that's a big market, and it validates my work on the way towards human. And the truth is, vaccine science doesn't require that sophisticated of assays. There are all these new fancy things like mass cytometry and things we could do. But the truth is, vaccine science has been around for a long time because the methods are relatively simple. The assays are taking serum and running microneutralization, which is an ELISA-based assay, or just binding. You can do RNA extraction and high-throughput sequencing, and we did those additional tests. But ultimately, the most critical question is the animal producing antibodies that neutralize the virus? And that's a simple assay to run. So I didn't need complicated things, and I didn't want to wait. I think my problem was that I would either have to wait I'm really appreciative of the Gates Foundation, but it took 3 years to get that money started. And if I had waited that long, I think that's fundamentally part of the problem of the whole pharmaceutical industry and academic industry, is that everyone's waiting for these grants. And so we have very slow progress. And also I was trying something that was pretty different. And the risk profile would seem high to someone who wasn't an extreme expert. They would look at this and be like, well, that's too different than what I know. So I'd rather only invest in things that I know. And so there was a gap, the biotech valley of death. And my way of solving that was realizing that Guatemala has animal facilities, and they have pig research. And I knew professors there. I knew specialists, animal care specialists, and my brother was a construction worker. So we just built a facility and we ran 3 rounds of studies, and we were able to run them very quickly. And I think that really helped us find the proof of principle and then do the iterative engineering to optimize the formulation.
That's no wonder you and your team kind of like racing for the queue. And so we know, like you just mentioned, you're developing the antibody treatment for what we are really suffering from, the coronavirus. And so we know that there's 4 to 5 other companies already working on such a antibody treatment. Do you think you will get this treatment out first?
I think we could. I think there's some other groups out there, and that's good, right? There's one serious competitor. It's the virus. We're all in there together. We're all working together to try to treat it, so that we can protect our lives globally, and so that we can protect the economy and get back to work and focus on creating new things, which is what humans are great at doing. And so I think that said, I think there are some very competent groups. My feeling is that Regeneron did historically very well with Ebola, and I think they're working well. And I sleep better at night, because if my medicine for some reason fails, I feel like their medicine could work. There's also a group called Upcellera that historically, I know the CEO, I like him. I think their group is competent. They took a patient that had survived and they searched through their B cells. I think it is likely that my medicine will have advantages and it will be faster. And so I've got some slides I'll go over, but in brief, what I did is, again, I asked, how can I make the medicine the fastest? I thought a vaccine is important, but it's not enough, because vaccines don't work very well on old people and on people that are immunocompromised. And this virus is the worst in those categories of people. And also, you cannot give a vaccine to someone who's already in the hospital sick. It's too late. They're going to be dead or recovered by the time the vaccine does them any good. So you need another treatment. If we were lucky, chloroquine or remdesivir or Kylletra would have been that treatment. But it looks today like if they're helping, they're not fully helping enough. They don't solve the problem by themselves. And back in January, I didn't have all those answers, but strategically, I thought it would be a good idea to start working on an antibody. So an antibody, it has the advantage that you can infuse it into a patient, like an antiviral, and it starts working immediately. Within 20 minutes, that person has protective immunity, and they'll have that for the next 8 weeks. And you're not relying on the patient's immune system, so it doesn't matter if they're old or immunocompromised, and everyone's going to get the exact same antibody, so it's going to behave in the same way in all of those subjects. Furthermore, the unique advantages that my technology has is that we have this ability to heavy-duty engineer the antibodies to have affinities that are far beyond what nature typically is able to deliver. And we're also able to increase the thermostability, so that the molecule is able to resist crashing out of solution, and we're able to concentrate it. And the reason that's valuable is that back when we were working on Ebola or other groups, they struggled because the original ZMapp antibodies were not very good antibodies. They precipitated, they crashed out of solution. You couldn't concentrate them very much. So you had to carry these very careful ice bags to keep them kind of cold, which is ridiculous when you're trying to deliver drugs to Africa. And so it was just impractical. What you want is a very thermostable molecule that you can concentrate like crazy, and it's resistant to temperature fluctuation. And that means you can do subcutaneous injection, so a little syringe rather than having to receive an IV, which is a significant advantage. It also is, just practically speaking, by September, reasonable projections are that we will probably need millions of doses of this medicine to go out internationally. And so if you can make a drug which is 10 times higher affinity, and therefore you can give 10 times less material to each person, a given vat of like a kilogram will treat 10 times as many patients. And that's important when you're talking about a major outbreak problem.
Yeah. So I'm very glad that you said just the other competitors. There's no competitor, only they're actually partners, and we have to work together. So this pandemic, unfortunately, is a global issue now. and infected every country. So from your perspective, how can global biotech companies work together to win this race?
Yeah, so I think that's been one of the things I found very gratifying. I've been invited on to a number of media stations. I think they bring me on to calm them down because people are freaking out about the economy or just their lives and their health. And what I tell them is this. I say, biotechnology has never been stronger than it is today. We were better with Ebola than we were with HIV. We were better with HIV than we were with measles, and we're better now. I've been humbled and impressed by how quickly the biotechnology community coordinated and shared scientific information like sequences, ability to grow the cells. And then personally, with my own project, I've had multiple companies just reach out and offer to work with me. So Abbott, to do additional screening and characterization, and they were interested in creating diagnostics. We had Charles River Laboratories to work with us to do the safety profiling. There's a company called Xencor that has mutations in the constant region of an antibody that increase its half-life. And they reached out and said, hey, are you interested in using this technology? There's a group, I'm not sure if I'm allowed to say their name yet. There's a group that lets us do very rapid scale up of GMP material that could save 7 months of work. And they reached out to us. And all these groups, I think it's easy to collaborate right now because we all want the same thing. We want to be able to produce a medicine that can protect our parents and our children and can make this no longer be such a medical crisis. As soon as we have a good medicine, it is no longer as dangerous if many people go to the hospital because they'll receive the treatment and they can go home. It'll be like antibiotics. Mm-hmm. And so for those reasons, it's easy to contemplate good business relationships where everybody benefits because we can all contribute whatever we are really good at. Let's put it all into the pot and we can make something powerful. And the goals are make the best medicine we can, to test it out in the most efficient way so that everybody everywhere can say, yes, this is good enough for my people and my population, and then produce enough of it that it can reach the world efficiently. And mm-hmm. that's how biotechnology can come together.
Mm-hmm. So have you been working with any Chinese company or organizations?
No, and that's why I accepted your offer to come, is that I think I need to. I think I'm in a position over the next couple of weeks where I'll know, yes, here's the affinity achieved, the neutralization. But my conversations so far have really been limited where I have a blind spot when it comes to all of Asia. I really haven't even been talking to Europe either. So what I don't want to be done, I'm worried that I will be stuck either not knowing about a technology that exists in Asia, a Chinese group that has some advantageous technology that could help here, or to be able to aid in distribution and manufacture, and to make sure I have not neglected to set up the appropriate trials. There's many things I'm concerned I may not be doing well, and I need to start planning to do those quickly, and I need partners to accomplish that effectively.
That's amazing, because we definitely, I know we have of course, most of the audience come from China, but we also have the audience from Europe, and actually have a very global presence. And I think you attract the global, the people to listen to today. And so I think I will stop here, and you will take over and to give the more the details of distributed biotechnology. And so after that, we will take the questions. I think I will reserve more time for the questions, because I know I already received the questions now, and I think people want to talk to you. Thank you.
That's great. Well, thank you, everybody, and thank you. Are you able to see my slides?
You have to share the screen again.
Okay, I'll do that. Share screen, boom.
Yeah.
And boom. And voila. Yes?
That's great.
All right, so hello, everyone. What I'm going to do is tell you, at least I think it's interesting, hopefully you'll also find it interesting, the story of our technology, why we chose an antibody as our method for targeting COVID-19, how we started the program, the theory on how we think we're saving many months of work, where we are, and what happens next. So what I'm showing you here is an infographic that I developed with SynBioBeta. This is a synthetic biology community run by John Cumbers here in the Bay Area. And what it's attempting to do is compare the relative merits of 3 different medicines that could be used to treat the COVID-19. The first is the repurposing of drugs like Ebola or HIV or malaria drugs. So remdesivir or Kaletra or chloroquine, respectively. And I know that the Chinese protocol, South Korean protocols, and now I think the United States is starting to include some of these medicines in those protocols. That's really, if a drug works well that you already have, that's your best approach because it's already been tested in humans and it's really just repurposing it. The challenge, of course, is that maybe those drugs aren't going to be that potent, and I think we're still determining that. At least in the US media, there's a lot of attention to the idea of a vaccine, and a vaccine is definitely important. But the problem with the vaccine is they're estimating 18 months before we'll have one. That's too far away. We need something faster. And the real problem with the vaccine is that you can't give it to someone who's already sick, because a vaccine, you give a shot and often you wait 3 weeks or 4 weeks, you give a booster shot. So then you're talking 6, 7 weeks before that person has time to build up a lot of protective antibodies. And that means that you can't give it to patients. You have to give it to healthy people. And that also means that when you run your phase trials, the human trials, you have to run these long, slow trials. So you have to give your drug to like 10 or 20 times more people than you think are going to get infected. Lots of healthy people. You have to give time for them to build up immunity. And then you have to wait and watch them for another set of 3, 4, or 5 months to be able to analyze whether the group who received your vaccine is statistically less likely to get sick than the group who did not receive your vaccine. So it's slow. That said, the value is that vaccines are a powerful platform. I love vaccines. I work on them in other areas, and they're pretty cheap to produce once you produce them. The cost of goods can be quite inexpensive. The middle ground that I'm going to talk about today is an antibody, a monoclonal antibody therapeutic, or a oligoclonal. So the idea here is that you get antibodies either from a recovered patient or a transgenic animal, or in my case, engineered from SARS, and then you infuse that into the patient. So it's like a vaccine, except you don't have to wait because the person's not making the antibodies, you've already made it. That means you can pick out a really good antibody. You can pick a picomolar, neutralizing, really solid antibody. You can give this to patients because it works right away to protect them. You can also give it to hospital staff, doctors and nurses to protect them for, it gives about 8 weeks, maybe 10 weeks of protection. It's more expensive. There's ways to make this cheaper. By default, antibody therapeutics in the United States often cost like $10,000 a dose, but that's a crazy cost because of what the pharmaceutical companies want to charge. The real cost of goods is it's really like a couple % of that. So there, in an emergency like this, you could produce large amounts of this and distribute it for $100 or maybe a little bit more. And then down below, I'm showing you the 3 different times it's going to take to be able to deliver a therapeutic, and I'll talk about that more in a bit. But the antibodies can be delivered faster because you can give them to patients for phase trials, which means you only really, within 10 days, you know, did the patient die or did they get better? So you have a quick answer, which means the trial is fast. And the other advantage is you can start running these things called a phase 1/2 trial, as they do in oncology, where right away you just spend more money and you look at 600 patients in your phase 1/2. And so you get safety, but you also get efficacy in the same study. And that means you really only need one study where you have efficacy before you can start going into your next study. And at the same time, you can release your drug through compassionate use or expanded access, which means that you can start handing out hundreds of thousands or millions of doses after one phase 1/2 study, which is the fastest route that I know how to think about to get medicine into people. All right, so I'm going to start jumping through some slides now. So a little bit of background about my company. As I mentioned before, we are computational immunoengineers. The company is built atop the ability to easily look at hundreds of thousands or millions of antibodies, and that could be in a person or in a pig or in an antibody discovery library. And I've got a bunch of papers you can take a look at that describe various algorithms I designed to help interpret the immune system and build better medicine using these kinds of tools. And that's really how we were able to launch our company without outside investment. The practical application, there's many cool things you can do when you can look at the immune system and read hundreds of millions of antibodies. But the blue collar useful thing you can do when you roll up your sleeves is that it helps build better antibody discovery technologies. So right when I built these technologies back in 2009, I immediately pointed them at phage display and at hybridoma. And I tried to ask the question, why do we not make more antibodies against the right epitopes that we care about? At the time, I was working at Pfizer, and Pfizer would give a hybridoma group and they would give a phage display group the same drug target, and the 2 groups would compete to try to produce an antibody. And both groups struggled. And I was always fascinated by why that was, because when I tried to run the mathematical calculations, I feel like the mouse should have hit the right epitopes more often, and I feel like the phage display libraries, there's something wrong with them. And when I started being able to deep sequence the libraries and then the animals, it became clear to me what some of those problems were. And that resulted in the next few years of my research, I was able to produce much better antibody libraries that had many more hits against drug targets. And that process really continued. I'm showing from 2009 to 2011, two published papers. 2013, you can see more hits and higher affinity hits just in a few years on top of a technology that had remained relatively stagnant for the previous 20 years. And so that ultimately resulted in the basis of the profitability of distributed bio. I think we've run 55 different antibody discovery campaigns in the last year. And part of our unique advantage is that we can create hits against really challenging drug targets like GPCRs and ion channels, HIV, anti-peptide MHC targets, and so forth. And it's partially as a consequence of us using something called superhuman technology, which is an antibody library that we computationally optimize to be about 2,000 times larger than historical libraries. And so because you have so many more shots on goal, you're going to hit goals more often. That's a unique part of our technology. So what I'd like to do now is just focus on the story of why we went after COVID-19 and how our technology enabled us to do so. So the story really dates back to some work we were doing in early 2019. We started a partnership with Peter Kim. He was the chief science officer of Merck and then became a professor at Stanford. And he came to us at one point and said, "Hey, Jake, I hear you have this awesome antibody library. I'm trying to make neutralizing antibodies against this new broadly neutralizing epitope on HIV. Do you want to partner up and try it?" And so I said, sure. And he came to us because other technologies had not successfully produced antibodies against that part of the virus. And we were able to do it very quick. In under 4 weeks, as a side project, we produced panels of neutralizing antibodies, broadly neutralizing antibodies that hit all the versions of HIV that they tested. And so we were pretty happy about it. And then we got a knock on our door from the military saying, "Hey, yeah, Jake, so we kind of like that technology because we're looking for a technology that could rapidly produce antibodies against a novel virus or a novel, whatever the zombie apocalypse strikes, we need some technology to be able to rapidly defend ourselves." So we said, great. We started engaging with them, and we've got really productive relationship with US AMRID, NMRC, RARE, and these groups. And we have a MTech grant around creating antibodies against some of the toxins around pathogens. So that stuff was already ongoing, and I was flying out in January 25th to Washington, D.C. to go to the biothreats meeting. This is a meeting that happens once a year where people around the world who are studying outbreaks and pathogens and also dual use, so potentially bioterror agents, can get together and talk about how to make better medicines to protect ourselves and better surveillance tools. And so I had flown out to go to the biothreats meeting and also to meet with DARPA around us applying our technologies to try to go after like hantavirus or Lassa or just random kind of some viruses that were of somewhat interest, but mostly it was just to basically act as a test run in case a real pandemic struck. But of course, the COVID was starting to break out during that period. So the meeting was ridiculous. Everyone was freaking out. DARPA had asked us to say, hey, throw all that other stuff out. Tell us what you can do for the new coronavirus. And I also saw Fauci. Dr. Fauci was presenting at it. And already in January 25th, he was saying, or that week, he was saying, realistically, we're not going to be able to contain this anymore. This is beyond China. This is going to affect the world. So he already knew at that point where this was heading. And taking all that together, I went up and immediately started setting up the plans I'm about to show you for how we can make an antibody against this extremely quickly. So here is some of the research. This is a slide I created in January, that week of 25th. What I did is I pulled down all the sequences I could find of the novel coronavirus, and I also pulled up sequences from SARS and MERS. These are all homologously related viruses. At the time, I think I found 19 sequences, so the world had done a really good job of sharing data even very early on and coordinating. And what I found is those early sequences were all identical. They had not mutated yet. And when I compared them, particularly I was interested in the receptor binding domain. So this is the top part of the viral spike. If you look in the middle, you'll see this kind of flower-looking beautiful thing. That's one of this beautiful but evil. That is one of the spikes of the coronavirus. And in the middle of it, you'll see something that has a bit of yellow and blue on it. And that's one of the 3 receptor binding domains. That's the thing that goes and binds to ACE2 receptor and causes the spike to open up, grappling hook in, attack the cell, and inject fuse its genetic material. So I pulled up those sequences, and they're shown at the bottom. And what I found is that MERS is very different from the coronavirus, the novel coronavirus. It's 19% identical. So 4 out of 5 residues has been altered. It's basically almost unrecognizable as the same protein. On the other hand, SARS is quite similar. It was 74% identical. So basically 3 out of 4 amino acids had not changed. This thing was relatively conserved. And if you see the yellow compared to the blue on the right, I'm zooming in on the receptor binding domain. And the yellow is positions that have been mutated between the novel coronavirus and SARS. And the blue has not altered yet. So these things are cousins. They're pretty similar. And that got me super excited because I looked at that and I realized this is actually an opportunity for us to go take advantage of years of research that was done on SARS to jump a molecule really quickly to be able to treat the novel coronavirus. And so here's what I did. What I did is I took advantage of existing structural data. So this is showing a series of different structures that recognize the SARS or MERS. And most of these are against SARS. And you can see the receptor binding domain. I've kind of blown it up in every direction, and I'm showing in the different colors the antibodies and where they all bind. And these had solved crystal structures, and many of these antibodies had 2 years of research on top of them. So we know they were neutralizing. We know they're effective at neutralizing in vitro. We know they protect animals when they're immunized. These would be amazing drugs if SARS ever came back. But the magic here is that what I was looking for is I was looking for antibodies that bound SARS in a way that I thought that site hasn't changed that much to the novel coronavirus. All of the sites have changed a little bit, but I have this technology called Tumbler that I could use to modify and engineer anti-SARS antibodies to also neutralize the novel COVID-19 virus. And by being able to do that, I could take advantage of a whole bunch of research on an extremely well-validated antibody with the right orientation and the right epitope and a known mechanism of directly blocking ACE2 and be able to adapt that to become an effective therapy against COVID-19. And if I'm lucky, the way I adapt it is I retain the neutralization against SARS. So I end up with a broadly neutralizing antibody, which is able to neutralize the novel coronavirus. It can neutralize SARS. It can neutralize many viruses in between. So these are our starting targets. So this is our process. And again, we do this commercially all the time. We do this for a bunch of companies. This is the super fast timeline that we don't normally offer to our clients, but we're able to offer it in a global emergency like we have now, where I had 2 team members. Sharad Daryakia was building out these antibody libraries. We have a technology that lets us make billions of versions of each of those antibodies I just showed you. And then Jack Wang was working with us, and he was engineering that receptor binding domain in various ways to be able to be displayed on magnetic beads and otherwise captured using his tags, Avi tags, and his tags, and as well as a TEV cleavage site. So all that work took about 4 weeks to go build all those pieces. At that point, we used magnetic bead manipulation robots, and we worked in shifts to conduct all the panning. Now, during this, when we were about to start this, there was the government shutdown here in California, but my team volunteered to come out anyway and keep working on this into the weekend, in the mornings, and the evenings. I'm just really appreciative and humbled by their dedication to this. And we were able to blast through the panning extremely quickly. That then gets us into using other robots for doing colony picking, robots for doing screening, characterization, and I'll take you to what the steps that are the next. But you can see here that this is a very fast path where we can produce molecules extremely quickly. I'll talk a little bit about Tumbler because it's a pretty sexy technology. So the idea is when I started deep sequencing antibodies from hundreds of subjects, I realized that the individual CDR loops of an antibody didn't really have that much diversity in them, and they were similar to what was found in other people, with the exception of one loop called the CDRH3. That was handy because it means that if I want to optimize a starting antibody, nature has already taught me, by looking through the blood of thousands of people, the kinds of diversity that can be populated in each of the other CDRs. And so we can use that information plus wet lab techniques to amplify out the CDR diversity from hundreds of subjects, synthesize some CDR diversity if we desire, and then recombine that on these fixed scaffolds. So this technology lets us make between 2 and 4 billion versions of each of those starting antibodies. So this says 500 million. We made the libraries larger than that. So Sharad made them about 4 billion. So you make a huge number of hits of versions. We then apply extremely vigorous selections. We have a population of billions of versions of those antibodies. And the idea is that here's an anti-SARS antibody. here billions of versions of it where we've modified it a bit, single, double, triple, quadruple, quintuple amino affinity changes, amino acid changes. And then we select them all against the novel COVID-19 receptor binding domain. And we ask which sets of mutants allow it to adapt successfully to now strongly recognize and neutralize the novel virus. We then narrow down that pool, the subset that recognize it, and then we apply computational methods to analyze that pool compared to the parental pool, and that teaches us which residue changes were causing the shift that we desire. And so then we synthesize the remaining optimal sequence. And you can read more about the methods I'm describing here in my Nature paper from 2017. So that's basically where we are. We thought the reason this was valuable, and my company was basically shut down for a period because of the shelter in place. So the entire company was focused on pushing this medicine forward. And I thought this was valuable, first because it could save many lives. If everybody gets infected, we're talking about over 200 million people that could die. And that's our family and our friends. And it's also, as a business owner, it's my employees. I want people to be safe so I can keep working. It also protects the global economy. There's a huge hit to the productivity that humans could be conducting if we weren't having to hide inside of our homes. And ultimately, I think we need a medicine to stop this. Otherwise, it's just going to keep slowly causing more and more small outbreaks. At the same time, I didn't want to just have a myopic focus on making one drug here. I was like, if we're going to do this, let's do it right. And let's not just win this one battle. Let's win the forever war against all coronaviruses. So by taking anti-SARS antibodies that were already extremely well researched and then adapting them to recognize the novel COVID-19, we are attempting to also retain their reactivity to SARS. So we have one medicine that will neutralize the novel COVID-19, it will neutralize SARS, and will neutralize things in between. And so that will put us in a better position if this virus mutates, or if SARS ever comes back, or if a new coronavirus pops up, we'll be much more likely to have a medicine that we can repurpose. There's also a marketing advantage here. If I can be the one to get to the molecules first, the world's going to hear about it, and I don't think my service business is ever going to have to compete again, because every company is going to be like, well, who should I do my optimization with? I know. How about the guys who saved this? So that's been, from a practical perspective, good for the business. And then the last piece was it was just helpful for my company. It forced us all to work on one thing, and so I think that's good for a business, to occasionally just have everybody focus and just look at one problem together and really nail every little piece to get precise. And so I think we benefited from that. So now I'm going to talk a little bit about timelines and partners. Let me see if I can slide this around so I can see it. Okay, great. So the week of March 30th, basically into this weekend and into next week, we're doing a whole bunch of screening. Abbott Labs Diagnostics is additionally doing 10 additional plates with us of screening, so we're able to look at more clones, because our goal here is we're trying to find the best possible mutant version of these molecules we've engineered, and we're screening a whole bunch of clones to help determine that answer. Out to April 6th, we're starting into the GMP process. So we're partnering there. We have Sutro has a cell-free platform that's GMP certified that can produce large batches of drug very quickly. And so we're exploring whether that platform would be compatible for us. And we're also talking to Ginkgo Bioworks about the possibility of using their platform to generate single drop-in CHO lines, stable CHO lines that we could grow up. And the idea is the Sutro platform would happen fast, the CHO would take longer, but it could produce more eventually. And those are the two strategies. In general, you'll see with my philosophy is always that if my army needs to cross a river, I'm going to build 3 bridges. I always have multiple strategies so I can afford to have a failure and still achieve success. And that's our goal of overlapping attack here as well. Around the week of April 13th, at that point, we're getting into potency. So we have this partnership with US AMRID from the Army. We're going to send over our leads, and they're going to test potency against the COVID-19 virus, as well as against SARS, that I definitely do not have in my laboratory, and I don't want to have in my laboratory. And the other advantage is I want them to be independent validators of our molecules. They can go test it, determine the strength of it, and they can report on it. And part of that is because I'm going to go shake the tree with the government and say, hey, you should go pay for expensive phase 1/2 trials and for the large-scale GMP scale-up. And so I want the military to be an independent validator of the potency of our molecules. Around by April 20th or so, we get towards safety and tox. And that's the partnership with Charles River Labs. They're a 16,000 person company, and they specialize in safety and tox. So we're really appreciative that they came out and offered to do that with us. We've been talking with Xencor, and we'll be characterizing the half-life of our molecules around April 20th in the same safety and tox studies. Xencor has a technology that they make mutations in the FC to extend the half-life by about 25%, which would be great, because then you're talking maybe 10 weeks of protection. And then that gets us in a position by July, late July, early August, of being able to run a phase 1/2 study. This is ambitious. This is an aggressive timeline. This assumes a red tape can be cut. And of course, it assumes that the medicine is potent. And I'm kind of pushing on all fronts to get BARDA or DARPA or some part of this large amount of resources mobilized by the US government to go pay for this study. At the same time, I need to be talking to the Asian arena. I need to be talking to Europe. I need to make sure that I don't want to get in a position of running this part of the way through and suddenly having some part of the world be like, hey, you can't send us your drug because we haven't run a study that is acceptable to us in an area that's acceptable to us. So I want that conversation to happen early so that we're well positioned to run all of these studies in parallel so that upon success, we can start releasing by September all the drug for expanded access or compassionate use, which is to say we can start releasing it to the world at that point. And I want to make sure we're not missing anybody. So that's the timeline. That is ambitious. This is about as fast as we can do credibly, and there may be places where we get slowed down. I struggle to imagine it going any faster than this, but of course we will try. It requires coordination of partners, and we're open to any one of these steps where we have someone who can come in and does something better than what we do. We want to talk to them because we all have the same interests at heart of being able to mobilize this to everyone. So that's it. I've left in the slides. If anyone's interested, there's been, if you're interested in learning more, I put a subset of the various media appearances and interviews. There's more stuff up on our website. I mean, if you really got a lot of time and you're stuck in house arrest, we've got podcasts and a whole bunch of other stuff if you want to listen to. And then we're making an effort from our team to be kind of a radical transparency where we are creating videos on a daily basis and just sharing them up on various social media platforms so people can kind of come check in, talk to our scientists and understand the progress we're making. Part of it is I just want people to understand what it's like to build a medicine and the good moments, but also the frustrating moments, and just understand the kind of the hard work that goes into doing it. And I'm trying to just make the laboratory very open so people have an opportunity to see that. And so with that, thank you all. That's basically where we are. I'd like to stop and then address any questions if there are, or have any discussions that are of interest.
Thank you so much, Dr. Glanville. I think you are probably the most like a transparent company, like open everything to the public. That's very amazing. So I think I do have a question. Like I know you are kind of like you and your team racing for the cure for the coronavirus. So is any of the resource can help you speed up the antibody treatment? I know you already said that you already did like as fast as you're doing as fast as you can, but since we have so many people here and but so many people are suffering, so I just wanted to think outside the box if any of the resource we can contribute.
Yeah, I mean, any ideas, I'm welcome. So what I'll do is I'll try to share where we are. I think the big pain points I've seen so far are, screening all the antibodies. We have partners here, but there are more opportunities. That needs to happen very fast because we're taking the leads. And my objective is I'm trying to find, if I can beat the best affinity binder, I'll use it, but it needs to be very thermostable, and it needs to be, I want 100 picomolar or better. That's a really high affinity antibody. And that's really an attractive one because my goal is to dose at 300 megs per patient, which is very reasonable if you have a high quality antibody. And that really enables a good thermostable antibody, and a high affinity means we could do subcutaneous injections, which is just so easy. You could go take all the doctors in a hospital and give them a little shot in the stomach, just the belly fat, or it could be a depot in the arm. So that's the first part. We have these neutralization assays. I think those are covered. The GLP and the tox, I think we've got that covered. GMP, we're looking, but again, if there's some other technology that could be faster, GMP is a major bottleneck for us. And not just, there's actually two parts to this. One is, can we have a technology in place? I think Sutro's technology is remarkable, but even if we have one, we're gonna have a capacity problem because they need to go produce much, we need access to biovats, and we also need access to some of their starting substrate reagents to be able to enable this globally. So certainly partners that could help build any of those, the production line of all the pieces that come together to manufacture, it could be a very large number of doses that need to happen very quickly. And so that is a human engineering challenge that definitely could use more resources and more help. I think if people have ideas there, they can talk to me, but I will likely refer them to Sutro if they're our appropriate partner, because they are definitely looking forward to this. And that was the conversation I had with them. They're like, okay, we can produce enough for your phase 1/2 study, but we need to think, what if it works? what if we suddenly have to produce 10 million doses? Like that's a challenge, and that means global coordination for capacity, and then a series of materials that they need to start. And then we're also talking about CHO that comes afterwards. There also be maybe technologies I'm not even thinking of that can help. And so I think just that whole conversation is something that is definitely of interest to us.
Okay, thank you. So there's a definitely, I think that we can actually, since we have so many people, we can definitely spread out the words and to check in the different region and to see how can we really speed up the process. And the next question is, this is the outsourcing, the discovery and the scale has been the trend for the pharmaceutical industry. So how do you compare it and contrast with the companies like the MAP and the Adacera and many others with less scalable and the platforms and the service? So actually, they wanted to understand if that's real possible for the potential partner subscribe or license all three platforms you have. It seems like your platform are very powerful. So that's the question they have.
Yeah, so we license out the antibody discovery platform. We have, I think, 10 companies that are currently licensing the platforms. And we do either services or we do licensing. And typically, if a company has the ability to do antibody engineering and discovery internally, they may license the platform rather than have us do the services. So both of those are flexible. We also license the software platform.
Okay, so that's good to know. And so there's another audience ask, how do you the results from the server virus neutralization assay to confirm the virus is fully neutralized?
Got it. So how do you run the assay? So the way those assays work, neutralization assays are, you basically take the virus, you take a substrate of cells that are susceptible to infection, and then you put a progressive dilution series of your antibody. So less and less antibody. Promise, I don't have the coronavirus. You
probably should take some water or.
Yeah, I probably should. Take it out of a pipette tip over here. So what you do at that point is you mix them and then you wait a little bit. And then the readout is really an ELISA. What it's doing is it's detecting the presence of nuclear proteins that are only produced if cells are infected. And so you run these things in replicates, you run dilutions, and then you ask at what level am I no longer seeing infection, which indicates like that's the dose that thing is still able to neutralize in vitro. Now and by the way, I know all those things because of Sarah Ives. She was my co-star in the Netflix documentary, and she's also my lead scientist for the universal vaccine technology, and she built up all those assays. So I know it because she taught me. So thank you, Sarah. For the missing piece here, which maybe is the question, is how does that microneutralization translate to in vivo? And so there, we can look back to SARS. So the antibodies that we're working with as our basis, those were characterized in some cases for neutralization in vivo in mice. And so we do know that these antibodies as monoclonals at a certain dose are able to achieve like 1 mg per kg would be okay neutralization and protection, animal protection, whereas 10 mg per kg was vigorous protection. And so we do know that there's a basis for this, that this does work, that these antibodies can protect an organism. In the same way that we know with Ebola, that achieved effective protection. We've seen with influenza that neutralizing antibodies can achieve effective protection. With RSV, there's an improved therapeutic that can achieve the protection. So this is a well-established path towards creating a monoclonal or an oligoclonal that can block up a virus and inhibit it from infecting the host. So we know that works. We have precedence in homologous related viruses that this will work. It's just really the question for here would be the first time we test that would actually be in a human in the phase 1/2 trial to see whether the ultimate question is, you give this medicine to someone who's sick, do they get better quickly or not?
Mm-hmm. So that's another question is because they're very interested in learning your computing immune technology engineering. So it makes the like a discovery, the new target much faster. So how fast it can be? Like
so yeah, I mean, this one's an example about how fast we can go. So this is a project typically for clients. If we're doing novel discovery, we quote 3 months to do discovery of a single chain FV. It would be another 2 months. We actually work with GenScript, so we outsource our IgG reformatting to GenScript. They're a great company. We do all the initial discovery internally, and then if we do affinity maturation and optimization, that could be another 3 months on top of it. or 4 months even. So here, obviously, we did not spend 6 to 8 months. We went boom, we did it in a couple months. And so that's really what happens if our whole company focuses on really crushing the problem. So here we spent about a month. I'll actually go back to this. No, we don't need the slide. About a month doing the designs using computational guided methods to design what we were ultimately going to order, which is all the DNA oligos and the frameworks and stuff. Then we stitch all that DNA together. Took the first 2 weeks for the designs to be designed and to ordered. And then it took a couple weeks for us to stitch all the DNA together, put the bits in place, a little bit more than that. And then after that, we go into the panning and screening. And if everything runs smooth, we were able to finish that basically in 2 weeks. And then at that point, you're into converting things to IgG. Normally, we go and we let it happen we let basically 4 weeks go by where we send it out to GenScript and we forget. And eventually the clones come back and then we transfect them. And you know, we're working on multiple projects, but here we decided to go super fast. So as soon as we have them, we're going to partner with the Sutro technology, and they can go produce these things in like 3 days. So it's cell-free IgG production.
Mm-hmm.
So that's about as fast as we can get. The computational aspects where they help is they help in the original designs. We have software tools to help us analyze the antibodies when they emerge, and we have software tools to interpret where the best theoretical sequence is. So if you have millions of sequences, you select down a population that are good binders. You can compare these two populations using statistical methods. So these are, you know, chi-squared log sums of amino acid shifts on a per positional basis. And that will infer where the trajectories in sequence space are, and it infers a theoretical sequence that's better than any of the ones you pulled out. That's sort of the perfect sequence. It has all the combinations of good beneficial things that you've selected for, and you're combining them into the perfect sequence. And so we test those. That's what showed up in that Nature paper, was a method of testing those perfect sequences. So not only do we pull out the good ones, but we can infer what might be the best. We synthesize them and we test them. We synthesize typically like 10, maybe 5 or 8 of them will be really, really good, and some of them won't work. So it's prediction. But that process can save you a lot of time because you're no longer having to go do everything in the lab. You're actually computationally inferring what the right answer is. But that said, we don't do all computer. It's a mix. It's a hybrid model where you use the computer to adjust frequency distributions, but you actually run the wet lab experience using magnetic beads and you know, wet things like proteins. And then at the end, we do analysis to infer the better sequences. And it's really, I think that hybrid model is where the future of biotechnology lies. I've always been interested in efforts to do entirely computational guided antibody discovery. I just don't think it's there. I think the scoring functions are not good enough. And I've tried this. I've taken antibodies of a crystal structure against a target, I've peeled them away, and then I've given it back to the program, and it doesn't do a good job of finding the right answer. And so that tells me that those scoring functions, like we are not in the near future at risk of losing our jobs in antibody engineering because the software just can't tell the right answer from the wrong one. And so I think you gotta mix the best things that computers can do and the best things that hands can do, and that's where you're gonna achieve success.
Yeah, that's a very good point. Actually, we still need to teach the computer what to do, right? So how much input does the partner have in using the platform and a database to search and identify and select a series of potential hits, other candidates, antibodies?
Sure. So if they were coming to us, they would tell us the target, we'd engineer it, we'd produce a series of hits, and we'd typically give them back 50 leads, or we could give back 100. If they were licensing the platform, then they can do whatever they want. They can search it however they want. We share the protocols with them, and they just do all of their discovery on their side. So they're unfettered in that respect. They can search whatever they want. The caveat there is that there's 76 billion, 7.6 e to the 10, a huge number of antibodies in there, but it is finite. So it's theoretically possible that they're searching through it and someone else is searching through it, and they could find the exact same antibody. So that's a caveat to be aware of.
Okay. So that's great. So next, actually, Dr. Shang wanted to ask you a question and also share with you some of the insight about China, maybe potential their opportunities to work with some of the potential partners. Okay, Shang, please go ahead.
Yeah, thanks. Thanks, Dr. Jacob. They did a really good accomplishment their work. And actually, I saw some other audience that have questions, but I really have the questions. I have one question is that the first part you have already have the powerful Supreme 3.0. But in the future, if you want to improve more, what would you like? In which way you will improve? Like the algorithm or the database or the other areas? That is the first part. And the second part, I think, is if you want to cooperate with the other partners, which kind of the partner you would like to cooperate? Because in China, we have many scientists, many companies. They have collected a lot of data, or they are good at the clinical trials, or they have the manufacturing abilities, or they have both have the big market. So that is my question.
Yeah, great, thank you. So I'm always wanting to improve. So Superhuman 2, we're creating a new library called Cosmic. The Cosmic library, what I've done is, Superhuman 2 works quite well, but we analyze it before and after selections. And so when you analyze it, even if it works better than before, I still see defects. So we're going to add in some new frameworks for antibodies that I think are getting more popular. I'm dropping one of the frameworks that I noticed. There's 16 frameworks in the library, and there's one of the frameworks that's kind of underperforming, so I'm going to kick it out. We're going to take advantage of new methods for making DNA to be able to. I want natural CDRs, so that the CDRs, the loops, the DNA comes from the blood of hundreds of people, and I want to continue to do that because the human B cells do an amazing job of selecting for the antibodies that fold up properly, and I want to take advantage of what nature has given us in our library design. But because I've sequenced so many of them, I now have learned the rules of how nature has made a bunch of these, and I can start synthesizing some of them and adding them to the design as well. And in the process, that's attractive because I can eliminate certain types of biochemical liabilities. So these are amino acid combinations of residues that are undesirable in a therapeutic, and I can reject them or deplete them from the library as I build it. So that's the library work itself. We're also building a CAR-T display library technology. So this is a very powerful technology for rapidly finding agents that could target cancer, as like CAR-T. So we're doing some testing on that, and I have not yet built, but I'm going to build some new platforms that are based on cellular engineering and cellular display to help me discover better bispecifics, BiTEs, and CAR-T targeting moieties. In general, I like the CAR-T principle, but it's very expensive. And so when possible, I prefer a BiTE or an IgG bispecific as an alternative platform that could bring two cells together to perform the attack. But we have clients that do both, and we have internal interest for both. On the software side, we're developing a software called Architect. And the idea is that it's just going to improve our methods using now formal machine learning to take large sets of sequences, a selected population, and infer the theoretical optimum. And again, I'm practical. Again, I want my army to cross that river. I built 3 bridges. So I'm using machine learning on one hand. On the other hand, I'm using some relatively simple math that I designed. It's a scoring function, which is really just it's a log sum of chi-square values of amino acid positional changes on each position, which then gives rise to a value, which is the net selection pressure on a given sequence given a selection framework. And that actually works pretty well. So it's my baseline, and I'm like, okay, if machine learning works, it better work better than that. Otherwise, it's dumb. And so we're asking that question now, and I don't know the answer yet. So those are my areas that I'm trying to make the technologies better. I think it's always good. You should always look at your technology and ask how it could be better, because first off, it's fun, and second, that way you don't petrify, you don't become part of the past, you're contributing to the future. In terms of partnerships, yeah, a lot. We are shifting increasingly into a platform of assets. We were characterizing preclinical, and then we were also interested in going into clinical research. And so both to have support in the preclinical arena as well as the ability to run trials and access a very large market, I need help. I don't have contacts. I really don't understand all of the cultural and business norms, and I don't have access to that market. And it's a large part of all of the world. So that is an area. It's part of the reason I'm here today, is to try to make some friends and begin to establish those sorts of connections that I anticipate I will increasingly need over the next few years as I build new medicines.
Mm-hmm.
Yeah, got it. Yeah, thanks. Got it. Actually, in China, we in the recent years, we have a lot of companies that they raise money. They have been the unicorns in the industries. And I guess you must know some of the companies, they are good at like the AI, actually, but they are in the different industry. But I think they have the similar algorithms. Each one will have their best algorithm, but you can cooperate. And also in the China industry, just China biotech industries, they get progress a lot, and I guess we can do some help.
That would be great. Yeah, I appreciate that.
Yeah, because of the time, I think we will take the last question. I think this is from the audience. They sent it to hear the first question. Actually, they said that you mentioned about the crowdfunding, and they ask, do you plan to do that again, like or now?
Well, here's my strategy around that. So my feeling is that for the phase trials, I really think there's a lot of money right now in governments, and I think governments should support that. They should pay for the GMP and they should pay for the phase trials. And so that is my preferred method. So before I go passing the hat, I'm really pushing hard on DARPA and BARDA in the United States, and the US AMR has been a great partner. And I'm just like, guys, you have so much money, give me some. This is the right, run this study. And don't not run others. They should also build multiple bridges, right? They should pay for this study and they should pay for a couple other studies for a couple other technologies. Because if I were running government, I would want to make sure no matter what, I have a medicine in place as soon as possible. And I think the world has the similar interest. That said, we have been reached out to by a huge number of people after the Netflix documentary, and then also from the recent media updates and around this project, offering to provide resources. And so I think there's two categories. There are large-scale investments, so people are interested in investing over $250,000, maybe up to a couple million, and we're considering a small syndicate of private investors for the company there, and we're starting to talk to them. And then for the folks that have a little bit of money and they want to contribute, we are considering creating a donate button and providing that for everybody. But again, I would rather take the government's money first, because you've already all given money to the government. Let's use that money. That's what that's there for. But at the same time, I think this is a fallback position, and I appreciate the support in case government moves slow, because government sometimes moves slow. So it's something we're considering. We're probably going to have a button up on the new website when it gets released. But I still feel like the government should pay for this. So I'm pushing as hard as I can for that.
Thank you so much. So I know we have taken a lot of time from you today, and it's very amazing. And also thanks for you to share details of the technology and what you are looking at to improve and the partnership. I think that the audience have been learned so much, and we can take from there. And also, as MSQ is really focused on bridge the Western and the Eastern, the biotech industry. So we definitely will continue the conversation offline.
Thank you very much for having me on and for all of your time this evening. I really appreciate it, and I hope we get to have this be the beginning of a larger conversation.
Thank you. And thank you and your team work so hard to keep us safe. And you.
All working away over here.
I want to say hi, everyone, and thank you so much for working so hard. That's very inspiring.
All right. Thank you very much. You have a good night.
Thank you.
Thank you.
Bye. Thanks everyone for joining us tonight or this morning. And so we will share the Dr. Glanville's presentation later. And if you have any question, and you can reach out to us, or you can reach out to Dr. Glanville directly, and we definitely will facilitate how can we speed up the process and find the reason for the queue. And I think we are together, we are going to win the race.
Yeah, absolutely.
With the scientists like Dr. Glanville. Thank you. Thank you again.
Have a good night. Cheers.
Thank you so much, everyone, for joining us. Stay safe and healthy.